Welcome back, everyone. What a great conference we've had today. Let's welcome our next presenter. We have Regen BioPharma, Inc. It trades on the OTC pink sheets under the symbol RGBP and RGBPP, and is focused currently on mRNA and small molecule therapies for treating cancer and autoimmune disorders. Please welcome its president and CEO, David Koss, and chief scientific consultant, Harry Lander. Welcome, gentlemen.
All right. Thank you very much.
We're happy to be here. Take it away.
Okay. So I'm David Koss. I'm the CEO of the company. On our slide deck, the first slide I'd like everybody to take a quick peek at is our safe harbor slide. As a public company, we're required to disclose the safe harbor provisions. And then we'll go on to slide number 2 or 3. Regen was founded in 2012 by myself and Dr. Thomas Ickham. We're a fully reporting company that trades publicly. We have a common stock that trades under the symbol RGBP and a Series A preferred stock that trades under the symbol RGBPP. We're focused on cancer immunotherapy, gene silencing, mRNA, shRNA, with a focus on checkpoint medicine. We have one investigational new drug application that has cleared the FDA, and we have 2 that are pending. We have 3 products in our preclinical development stage. We have a focus around the checkpoint NR2F6, which seems to have a great deal of influence on modulating cancer tumors. We have 8 granted patents and 17 additional patent applications that are pending. The company itself is fairly streamlined in nature. This is because we do our research through contract CROs. There are contract research organizations called CROs. This means that we don't have to use our own labs to do the research. We have scientific consultants that work with the company, and this makes for a much more parsimonious use of funds as we're moving forward. Our actual commercialization strategy is to get through phase 1 and 2, and then form some sort of partnering relationship with a large pharmaceutical company to move through phase 3. Next slide. Oh, let's see. The current management team really involves myself, and Harry Lander is our chief scientific consultant. But we have a very large advisory board. Just a second here. So for some reason, oh, I see. Pardon me, this is a new experience for me, so I'm trying to get up to speed. Okay, so slide number 6 is starts our scientific advisory board. You can go on to slide 7. More. And then, well, let's see, slide 7 takes us into our pipeline. And I will hand it over to Harry to discuss the drugs in development.
Thanks, David. And thanks, everybody, for letting us present our science. I think we have an interesting pipeline at Regen BioPharma. We have a series of drugs that are in development that are range from ready for phase 1 testing. That's what we call Heme Accelerate. We have 6 programs on the left that you can see. 2 of them, as David mentioned, the INDs have been cleared. Most of you know what an IND is. Just to be clear, when the FDA requires an investigational new drug application, an IND, to be submitted and approved, or what they call cleared, before they allow you to conduct phase 1 trials, the very first of typically 3 phases. So it's a big deal for a biotech company to get an IND cleared, as we have for Heme Accelerate. We have 2 that are in discussions with the FDA, and we have 3 interesting preclinical programs, Diffron-C, Chimeraptor, and Duracar, which are focusing on different elements of this transcription factor, which is a major focus of the company, but not the sole focus, which I will describe in some more detail on some following slides. So the key is that the first 3 programs, which are, one is cleared for clinical trials, 2 are almost ready, are cell therapies with different targets within them. And then the last 3 are both cell therapies and small molecules or biologics that can attack this NR2F6 molecule that we'll talk about. So I'd like to go through each of these at a high level, and just to give you a feel for the science and how we're approaching the development of these drugs. So Heme Accelerate is a stem cell based product, and it's used to treat bone marrow damage. Bone marrow can be damaged for many reasons, for chemotherapy or radiation therapy for cancer patients, aplastic anemia for people who have a rare genetic defect. And so in other cases, bone marrow can be damaged from radiation damage, not just from treatment of therapies, but from, for example, a nuclear war. In theory, you would get a lot of radiation damage. So that's, and many countries are prepared for radiation protection. So Heme Accelerate is an interesting approach. We have, as I said, a drug. We're ready to start our phase 1 clinical trial using Heme Accelerate. D-Cell-Vax is a different type of cell therapy where we take the dendritic cells, one of the immune cells, out of the body. We silence one of the enzymes within the body using our patented approach, which is gene silencing of indolamine dioxygenase, IDO, one of the many enzymes within the cell. But inhibiting IDO releases these dendritic cells to become very active in presenting antigens to the rest of the immune system. So it has a way of activating the immune system. And this is a very well-known target, IDO. And in fact, Bristol Myers Squibb bought Flexus years ago for $800 million. Again, and Flexus was focused on this IDO target. And there are many clinical trials now focused on IDO, but not in dendritic cells. So it's a kind of a novel approach. The manufacturing process, I won't go into the details about D-Cell-Vax manufacturing, but it's relatively straightforward. It takes about 6 days. And if you compare it to the CAR T therapies, which many of you may be familiar with now, which have sort of transformed cancer therapy, and I'll talk about that in a bit later. But a typical CAR T therapy manufacturing, it takes about 3 weeks, 4 weeks, depending upon the company, and is very, very expensive. It could be $400,000 for a single treatment of one of these CAR T therapies. So our manufacturing process is far more simplified and far shorter, which has a lot of advantages. So those targets was indolamine dioxygenase and stem cells. But now a big focus of the company has been on NR2F6, one of these transcription factors that is a checkpoint. Checkpoints are proteins or transcription factors that keep the immune system in check. That's why they're called checkpoints. There are now drugs that release the immune system from their checkpoint. They're called checkpoint inhibitors. So you have to inhibit the inhibitor to release the T cells or immune cells to become very active. And there are billion-dollar drugs on the market, as you know, anti-PD-1, anti-CTLA-4, that are checkpoint inhibitors. So NR2F6, there's no known target for it. It's a very interesting transcription factor, but it's well understood to be a checkpoint. It's been, I would say, very well published around that. We have patented approaches to inhibiting NR2F6. And so several of the drugs that I'll talk to you about now focus on inhibiting NR2F6. And in fact, T-Cell-Vax, one of the drugs we have an IND filed with the FDA, is where we take the T cells out, or the immune cells out of the cancer patient. We silence the NR2F6 in those cells and then reinfuse them back. And the idea there is that you would, again, release the checkpoint and allow these cells to become very active. And here the manufacturing process is very simple. I mean, simple as far as cell therapies go, but it's one day. So it's a very brief process compared to the CAR T therapies that I mentioned. DuraCAR is something we're very excited about. So here we're taking, the idea is to take existing CAR T therapies. So CAR T therapies are therapies where patients' immune cells, cancer patients' immune cells are removed from their body. The T cells are modified, and a protein is put into them that recognizes the patient's own cancer. And then they're reinfused back into the body. And this CAR T therapy, which was first started in 2016, has become extremely successful at curing certain cancers, particularly the liquid tumors like B-cell lymphomas or leukemias, and has a very high cure rate, long-term durability. And it's been wonderful. The problem is that for solid tumors, which is the majority of cancers, they don't have a major effect, maybe on some lung cancers, but in general, they don't have an effect. And so that's part of the holy grail for cell therapies, is to create CAR T cells that can treat solid tumors. One of the major reasons that CAR T cells do not affect solid tumors is that the solid tumors have a microenvironment around them. They create this environment around them. And in that environment, they secrete molecules that shut off the T cells. In other words, they activate the checkpoints. So in our approach, we're actually silencing NR2F6, a checkpoint, within the CAR T cell. And we can do this very, I would say, elegantly, because the CAR that's put on the CAR T cell, we just add the shRNA. So we use the same molecule that we express the CAR in the T cell to also express this inhibitor of NR2F6. So we have a good, I think, a strong patent filed around that, and we already have a patent approved around the shRNA. So we're excited about moving that forward. And it's currently, we're working with CROs, as David has mentioned, to move this program forward. Differon C is another approach to using NR2F6, where we would actually take the siRNA and inject it in vivo to inhibit NR2F6 in your body. And there are approaches to using siRNA in that way. There are several ways, and we have some thoughts. We're focusing right now more on DuraCAR, but this is something that could be very interesting as well. Again, patent protected. And chimers, so aptamers are very interesting. They're short pieces of DNA or RNA that act just like antibodies, so they can bind to various proteins. And what we've done is we've designed aptamers that, again, are bifunctional. They bind to proteins that you want them to, let's say T cell proteins or tumor proteins. And to it, we have linked this RNA that inhibits NR2F6. So we're delivering the RNA directly to the cells in the body specifically. And so that's a very nice, and that's a platform technology, because we can target many different cells and different approaches using this. And it's actually not a very expensive approach. It's a nice way to approach it. And then finally, we do have a small molecule program that's been going on for a while at NR2F6. And this is small molecules, as you know, those are more traditional drugs like aspirin or the cholesterol lowering drugs that I take and many people on this call take. Those are small molecules, and traditional, we've looked at identifying small molecules that inhibit NR2F6 or activate it for treating autoimmunity. And we've identified some, and we just recently had a patent granted around some of those structures. So that's also quite interesting, because no one has yet published small molecules that actually inhibit NR2F6. It's one of, as I said, it's one of these. It's called orphan nuclear receptors. No one really knows what it naturally binds to in the body. So it's quite interesting. And as David mentioned, we have these 8 granted patents, not all around NR2F6, but some around other approaches, because again, we're always trying to diversify risk and diversify our approaches. And we also have, of course, these 17 patents that are pending, again, around both mRNA approaches and our 2F6, but also like adolamine dioxygenase, which I mentioned before, other targets as well, not putting all our eggs in one basket. But we do have, I feel, a very strong patent portfolio that represents composition of matter as well as methods. So I'm going to hand it over to David to close out the presentation.
All right, thank you, Harry. So the key points to take away from our presentation are: one, Regen has a very strong product pipeline. We are very much involved with immunotherapy and gene silencing. Two, our approach to therapy development is centered around this idea of conducting phase 1 and phase 2 of clinical research, and then forming a relationship with a large pharmaceutical company after that to carry the torch through phase 3, which typically will be geometrically stronger, more costly than phase 1 and phase 2 combined. Our exit strategy is actually pretty simple because we're a publicly traded company, so you can get out of it whenever you want to, but the inflection points potentially are going to be times when we are finished with phase 1 and 2, and we form a relationship with a large pharmaceutical company. We would expect an inflection of cash, a large sum. The company would be then in position to both reinvest money in research as well as distribute money to the shareholders. These are the key strategies in Regen. I think the fact that we have kind of designated ourselves as a virtual biotech company, meaning we don't have a laboratory of our own. We're using these contract research organizations, which allow us to move as fast or as slow as we need to based upon the environment we find ourselves in. I thank you very much for listening to our presentation. I look forward to taking a few questions here. And with that, I'll turn it back over to Anna.
Great job, gentlemen. Thank you for this exciting presentation. Can you tell us why you think the CAR-T approach, which you call DuraCAR, is so important and why it's going to work?
Harry, do you want to take that?
Sure. Thanks for the question. Yeah, it's interesting that someone would focus on that. So, yeah, we're very excited about DuraCAR because, as I mentioned, so the CAR-T therapy is an exploding and very exciting new way to treat cancer. And in fact, they're starting to think of ways of using CAR-T cells to treat other diseases outside of cancer. But as I said, in our view, to get to solid tumors, you can't use the existing CAR-Ts. They have to be supercharged. And we believe that by inhibiting NR2F6, based on the scientific data and the data we're now generating, we believe we will be able to supercharge CAR-T cells and prevent this ability of the solid tumors to turn off the T cells.
Great. And I see that you have several very exciting potential drugs in your pipeline. What are they and how do you prioritize them?
Well, if you go back to the slide on our pipeline, you will see the drugs we have. The key things for us are looking at the niche markets and what they represent, excuse me, both financially and addressing areas that are currently underserved. So if you look at them, the priorities are going to be the D cell vax and the T cell vax and the DuraCAR, We expect those three to be very important for patients, and we expect them to be very profitable for the company and ultimately our shareholders.
And you seem to have many patents granted and filed. So do you plan to keep filing additional patents?
Yeah, our goal really is to protect our science, our intellectual property. And the best way to do that, as we discover things, we want to file patent applications that protect everything that we have identified and concluded to be important in the directions that we're going.
And let's talk a little bit about your streamlined management team. Do you plan on beefing up your team? Tell me a little bit about that.
Yeah, right now we operate a very lean management team, which utilizes a lot of scientific consultants. My goal is to raise some money, more money for the company that would allow us to bring in key members of the management team that would potentially allow us to move things even more quickly without getting into a huge overhead like so many biotech companies do. A lot of biotech companies go out, they raise enough money to buy a building or lease a building, equip it with all kinds of things, and hire lots of researchers, clinical researchers, and then they find themselves in a position that they can't maintain that situation, and they end up going out of business. In our situation, we're structured by virtue of being this virtual biotech company that employs contract research organizations. We don't have the initial overhead that a lot of companies have, but it is still our goal to bring in key people for key situations that will help the company move forward as it grows and matures.
And last question due to time, do you plan on developing your drugs all the way to market, or do you plan on partnering with a large pharma?
Well, I think as I've outlined earlier, our key strategy is really to get through phase 1, which is the safety aspect of research. So you don't want to harm any patients. Once you've gotten through that, you move on to phase 2, which is all about efficacy of your therapy. We want to be in a position that we demonstrate that our therapy doesn't hurt a patient and that it does in fact have a level of efficacy in treating a certain illness. Beyond that, the cost factors in going from phase 2 to phase 3 are geometrically larger than any of the previous research. In forming a partnership with a large pharma, it puts us in a position that we are not forced to A, raise the money to do the phase 3, and B, in a position where we have a high risk associated with putting all the eggs in one basket to perform clinical phase 3. So it's our goal really to show the value of the therapy and entice a large pharma company to come share in that responsibility with us, of which we expect to generate a great deal of revenue for the company.
Wonderful. Well, due to time, we can't take any more questions. We've got tons of questions for you, gentlemen, and we'll send to you so you can answer directly. But do you have any closing remarks?
Well, I'm very, very excited about being in a position to share the Regen's story to people that haven't heard it or that have heard a little bit about it. As we have developments that come up, I'd love to come back on your program and go ahead and share those things with the audience.
Yes, please come back and join us again.
All right.
Thank you.
Thank you very much. Appreciate it.
All right. Thank you both. And thank you, everyone. We've had such a great conference today. Thank you for joining us, and to all of our presenters. Remember, in just a second, you're going to be redirected to our registration page for our next conference. Be sure to stay on to reserve your spot early. On behalf of all of us at Emerging Growth, I'd like to thank all of our presenters and attendees for making this such a successful conference today. Remember that a complete replay of this conference, separated by company, will be available on the Emerging Growth Conference YouTube channel, youtube.com/Emerging Growth Conference. Subscribe there and follow us on Twitter at Emerging Growth C. We always post new information on Twitter first. I'm Anna Berry, and on behalf of myself and our entire team who made this event possible today, we wish you a successful, healthy, and happy rest of your week, and we'll see you again very soon. Thank you all.